Okay, guys, so now here it is. This is my favorite chapter of all of the chapters. So this is dealing with metabolism, essentially. This is where I like to ask my students, since you guys are online students and I get to interact with you as much about this as much as I would like. I don't get to ask you that question of like, how many of you guys know, why we breathe in oxygen and, why we exhale carbon dioxide, because I'm about to tell you why. But most people think it's because, because the red blood cells, they pick up the oxygen from whenever we breathe in and then carry it to the tissues, and we exhale carbon dioxide as the waste. And why? So why do you need oxygen? Why do you exhale carbon dioxide? Why those things? And why exactly, like, where in your body is this even happening? Like, why do you need it? Like, where in your body is this going on? Why do you have carbon dioxide as a waste? Why any of that? So this is that answer. And I love this. And this is the reason I'm about to explain to you the reason why you breathe. And it's just incredible to me. So anyways, let's start off with the general concept. I can't believe there's just this little amount of words on here for this summary, but it is what it is. I say that a lot. Making energy. So catabolism. Cats are awful. I don't like a cat. I'm sorry. I know you guys probably like a cat. I don't like cats, but they are pretty destructive, right? I think we can all agree on that, whether you like them or not. They tend to be a little bit destructive, whether they claw on your furniture or whatever, or you have to go through the effort of teaching them to claw on a claw post, scratch post, or whatever it is. They're destructive. That's just how it is. Okay. Cats. Catabolism. Cat. These are breaking things down, breaking things apart, tearing things apart, okay? So we're going to tear things apart so that we can store the energy that is released from tearing it apart. That's the only reason I'm bringing up cats, is so that you guys can remember catabolism, tearing things apart so we can store the energy that is released, okay? Anabolism is the opposite from that. Anabolism, we are making larger molecules. So we need energy to do that, which is why catabolism has to happen. Catabolism, we're going to break things apart so we can store that energy so that we can put bigger molecules together where we need bigger molecules. Okay. So that's metabolism. Metabolism is basically all of the chemical reactions that are going on in your body, often, pairing catabolism with anabolism to power one another. So enzymes, these are catalysts, essentially. So a catalyst is anything that helps lower the activation energy of a reaction without being used up as a reactant itself. Yeah, so it's going to help a chemical reaction go faster and better by changing its shape, along with helping the actual chemical reaction go on, but it can be reused over and over again. Whenever an enzyme, that organic catalyst that we're using, acts on a molecule to change it from A to B, that molecule that's acting on is called the substrate. A cofactor or a coenzyme is an organic cofactor. So you can have cofactors that are like metallic ions and stuff like that, like iron, for example. But basically, these are things that help the enzyme do its job. So it needs those things to help it function better. That's a coenzyme or cofactor. So a coenzyme is, again, the organic version. So it's important then now. So we have our enzyme and we have our substrate that it's going to change. And sometimes it needs help from a cofactor or coenzyme, right? So how do you control the function of these enzymes? Well, we can have competitive or we can have non-competitive inhibition. And we'll get into those here in a moment. First, we have the enzyme classes that you guys might want to be aware of. The oxidoreductases. This is transferring essentially electrons. The transferases, putting one group from one molecule onto another. The hydrolases, adding in water, or removing. So hydrolases versus the ligases basically are going to work in tandem with one another as far as adding and removing water in order to, break or form bonds, depending on So the ligases are forming the bonds and the hydrolases usually are breaking the bonds. They both involve water. And then we have the lyases. The lyases have to deal with double bonds, and the isomerases have to deal with shape, essentially. Like you could different shapes of a molecule, like a D form or an L form, or orientations of molecules, they have to deal with changing that up. So then we have denaturation. Denaturation essentially is referring to, you can take an enzyme or usually enzyme or nucleic acid or whatever it is, and denaturation is referring to breaking it down, not necessarily apart into pieces, but its shape is important. And we've talked about this with proteins, right? In chapter 2, we talked about how important shape is to function of proteins. And it has a lot to do with those hydrogen bonds and everything like that. Well, if you add a lot of heat, or you denature with a massive pH change or something like that, then you can completely destroy the structure of a protein, and therefore it cannot function. That's denaturation. All right, so back to competitive and non-competitive inhibition on how we're going to control the function of these. So if you want to destroy the ability of a cell to function, you might denature the proteins in that cell in order to kill bacteria or something. You can do that with heat pretty easily. So, and we'll talk about that in Unit 3 a little bit. But yeah, so denaturation is an effective way of controlling protein function, but the cell usually doesn't control it that way. The cell usually controls it through competitive or non-competitive inhibition. In competitive inhibition involves time where the inhibitor, whatever's causing inhibition of the function of the enzyme, that inhibitor binds to the actual active site, the site where the substrate would go, the thing you're acting on. However, if you, like, let's say, we say important structure of the protein, right? Let's say your protein looks like this. And this is your substrate, where your substrate binds. And now we've got a non-competitive inhibitor. It comes in, and then when it binds into its little spot, not at the active site, the regulatory site, not at the active site, the regulatory site, okay? Then this will change shape. So now this guy, the substrate, cannot bind there anymore. That's how that would work. All right. So we've mentioned ATP before. That's adenosine triphosphate. ATP is our energy molecule. It consists of a nitrogenous base, in this case, adenine. So don't worry too much about what all that means, because we will talk about that later. But adenine is like the A in the ATCG of DNA. Okay. So it's a nitrogenous base. It has a sugar. It is a 5-carbon sugar that is associated with it. That sugar is called ribose. Yeah, that's like ribose, like RNA, ribonucleic acid. So ribose, and then it has 3 phosphate groups. Those are highly negatively charged. Each one is. So as we have a negative, stuck to a negative, stuck to a negative, that third one on there really wants to pop off. And so we're going to pop that off later to drive chemical reactions. First, we have to make it. We have to pop it on. So that's what this whole process that I'm about to get into, y'all, is about. So I'm gonna tell you why you breathe. I love this. So, okay, aerobic respiration. Essentially, we have 3 steps here: glycolysis, the Krebs cycle, and the electron transport chain. In glycolysis, don't worry too much about these little details and understanding all of the little details on everything. It's not that important that you get these little details as far as like, oh, it went from glucose and then, glucose 6-phosphate. And I don't need you to know that as much. What I do want you to know is the following. We're going to break down glucose into a 3-carbon molecule. So glucose is 6 carbons, okay? In glycolysis, we are taking that one glucose molecule, that 6-carbon molecule, and breaking it into 2 3-carbon. Let's check. Where's this? I don't have my little pen pad. So we're going to have 3 carbons on those. So there's going to be 2 of them. These are called pyruvate. That's the point of it. Do we make some energy in this? Yes. I am not concerned about it, okay? The main point, the main take-home message with glycolysis, we made 2 3-carbon pyruvate molecules. Cool, okay. So pyruvate is going to get turned into another 3-carbon molecule called acetyl-CoA. And then it's going to go into the Krebs cycle. It's also known as the citric acid cycle because one of the intermediates, citric acid, okay? Not that important that you know that. I do really want you to know that in the Krebs cycle, what are we going to make? We are going to be breaking down those 3-carbon molecules. We're going to break them apart into byproducts of carbon dioxide, and in the process of that, electron carriers. So I'm just going to put electron for now because that's the important aspect of it. But this would be NAD. That will become NADH when it's carrying the electron, and FADH2. I'm just going to put this for now. I'll put both of them. Okay, so this is what's made in the Krebs cycle. Cool. So we've taken glucose and we've broken it apart into pieces, stolen out the electrons, and the electrons are associated with their hydrogens, by the way, that they came from. Because now we've got CO2, where did all the hydrogens in our glucose go? Well, they went into, some of it went into this. So this became NADH. That H is a hydrogen associated with its electron, right? It's not a proton, it has an electron with it. So that's important to remember. So already I've shown you where carbon dioxide, this is why you breathe out carbon dioxide. This is the only reason in your entire body that you breathe out carbon dioxide. No joke. So that's why you breathe out carbon dioxide. All right. So we've made NADH and FADH2, and you're probably like, a big whoop-dee. I don't care. Who cares over that? What is this even? Okay, fair. The H's, I've told you that they are hydrogens with electrons associated with them. You can see them like this. This is how we've been looking at hydrogens for a long time in this course already. These are H+, this is your protons, right? And then also associated with their electrons. The proton plus their associated electron. That's what these H's are. Okay? Look at them that way. Next is the electron transport chain. The electron transport chain, that's the big daddy, okay? It is essentially a whole bunch of proteins and molecules that we're going to line up and align and blah, blah, blah on a membrane. And what's going to happen here is that electron from the NADH or the FADH2, so the electron, the actual electron portion of it is going to be shuttled through the electron transport chain, UTC. Okay. And while that is happening, that proton that was associated with it, because we know they didn't come like alone, right? So it wasn't just electron, right? It came with the H. The protons are going to get moved from inside of the cell to outside. And so we are, as we go across this thing, going to shuttle more and more of these guys out. All right. So this is happening in the electron transport chain. So let's say we've done all that. We've got all of our protons out there, right? we've created a gradient. There's a lot of H plus positively charged protons essentially outside of the cell. They want to come back in to equal everything out, right? So it just so happens that at the end of all of this is an enzyme called ATP synthase. And what happens with ATP synthase is these protons can come back in only through ATP synthase. And it does exactly what it sounds like. We're going to go through, this is an oxidoreductase. So we're going to go through, basically chemiosmosis powered phosphorylation. And what we'll do is we'll have ATP getting phosphorylated into ADP getting phosphorylated into ATP. So all that energy being stored into that now energy-rich molecule. Right, great. So we're moving these protons back in from that proton motive force that we have created. That's that gradient that we made. Move those protons back in through ATP synthase, powering manufacturing of ATP, our energy storing molecule, right? ADP becomes ATP. Yay, we've got all this energy and great. That's not the end though. So I told you about why we exhale carbon dioxide. Where did I tell you that we inhale oxygen? And I told you I was going to do it, didn't I? Here it comes. So we've got all these freaking hydrogen protons in here now, right? We've moved them all back in. We can't just have them chilling. And what about the electrons? Like now we've moved them through this friggin' chain and we brought them to the end over here. What about these electrons? Well, don't worry, we're going to put them back in with their hydrogens. And the way we're going to do that is we're going to take the electrons and these two hydrogen atoms, and we're going to take a half of an O2. And all of this makes one H2O. And that's how we take care of that excess protons and clear them out. That's how we take care of those excess electrons that were flowing through the chain so we can keep more flowing through. So this is done by an enzyme at the end, cytochrome C oxidase. Just FYI, that's the enzyme that is associated with doing that in aerobic respiration. And you can test for that with a special testing drops that we'll use in the lab to look for oxidase. So that's what it's looking for, cytochrome C oxidase. And this function here. This, you exhale carbon dioxide because you broke down glucose or associated sugars in the Krebs cycle, and you needed to steal the electrons. Your mitochondria do that for you. Then in your mitochondria, you have the electron transport chain, and you shuttled your electrons through and you brought your protons back in and made your ATP. And now you got to get rid of those electrons and the protons. So you inhale oxygen in order to enable you to get rid of the excess so that you can keep making more energy. If you do not inhale oxygen, you will not be able to get rid of the byproducts here, and you will not be able to continue with making energy that is needed constantly for your cells just to stay alive. And that is why you die if I suffocate you with a pillow. That is the reason. Pretty cool stuff, I think. Don't think about the suffocating part, but pretty cool stuff, I think, as far as explaining like why you breathe. This is the reason. This is the reason that your lungs even exist. Crazy, all of this. So it's as simple as that. You just learned the chemistry behind why you have lungs, essentially. Why your body has donated so much of its energy and time to making sure you can breathe, because this is why you die if you don't. So crazy. I think it's beautiful. I hope you do too. So anyways, that's aerobic respiration. So just to read through everything that's written on up here, I gave my little explanation, my spiel. Glycolysis breaking down glucose into the 3 carbons, right? Going into the Krebs cycle, making carbon dioxide and electron carriers, things that literally are just carrying electrons. We're going to shuttle those electrons through the electron transport chain, create the proton motive force that will drive ATP synthase to make ATP. We need oxygen as the final electron acceptor. That's why this is aerobic. That's why we suffocate and die. This is electron transport chain where ATP is going to be made. This is where most of it's going to be made in all organisms. So in fermentation, they don't use oxygen. They just stop after glucose or glycolysis. And after glycolysis, and you get that pyruvate and everything, they use that essentially to work as their final electron acceptor. So they don't make anywhere near as much ATP. You make a ton of ATP in that electron transport chain, which is why it is so important and vital as far as keeping your body alive. Prokaryotes and bacteria, aerobic respiration, that's all going to be happening on the inside of their inner membrane of their cell envelope. Eukaryotes, like us, this is going to occur in mitochondria. Mitochondria do this for us. This is actually why this is some support in favor of the endosymbiotic theory, as far as we think that prokaryotes who do this for themselves and do this on the inside of their membranes, that some precursor cell back in the day took up a prokaryote that was able to do this, and we were able to harness the energy that they made. And that's where mitochondria came from, which is why they have their own genetics. Pretty cool stuff. So, fermentation, right? So we're going to break down the sugars and carbohydrates into carbon dioxide as a byproduct and alcohol or acid byproducts as well. So that's why you get fizzy beer, and it's why we get like kombucha and kimchi and all of these things that have that acidic sauerkraut, all of that. And what's going on as far as making cheese and yogurts and all of that, that's fermentation. So that's an alternate route of dealing with making energy where we don't have oxygen available in that electron transport chain. Okay. And then we have photosynthesis. We had mentioned that briefly before. We're using the light cycle to capture energy from the sunlight, essentially to power carbon dioxide fixation into glucose. So energy that we make from sunlight, ATP, just made similarly to how the electron transport chain works actually, but without electrons, we're using sunlight to excite things. And then they make a little bit of ATP so that they can generate glucose and then break glucose apart in aerobic respiration. That's actually how plants work. They still go through aerobic respiration, but they need to fix their carbon into an organic molecule like glucose in order for them to go through it. So that's kind of a neat thing too, a way around having to have the sugar available for you. You make it yourself from the air, the thin air. So cool stuff. So that would happen in chloroplasts in eukaryotes, whereas in prokaryotes that are able to do this, like cyanobacteria, this will all be going on the surface of their inner membranes. And that's that. So that's metabolism. I hope that was useful for you guys. Again, I think this is so friggin' fascinating. It's such a beautiful concept, and I hope you guys enjoyed it too. And I guess I will see you guys next time for genetics.